Okay. Yes. So thanks for inviting me to and for the opportunity to present this work, in which, as Stephen Smith gave a little bit a kind of a gist of it, we are going to try to understand how best to interpret brain age in no differences in brain age in cognitively healthy older populations. So super. So what is brain age? I think most of you already know, but still I'm going to repeat a little bit. Essentially, brain age is a measure in which we estimate by applying machine learning to a series of MRI features to estimate chronological age. Here you can see a typical pipeline. We often use, although not always, we often use like two data sets, one for training the model and one test data set of interest, in which we apply the model and get a brain age for each individual. The difference between brain age and chronological age, the residuals, are like 99% of the time the measure of interest, in which we correlate it with given outcome of interest. So this is a quite popular and quite generic measure, that the residuals that are often interpreted as a biomarker of individuals' brain health. So, and in the last few years, aging research has repeatedly shown how higher delta, yes, we use delta, it has, like, the residuals have different names. It can be, we use here delta and back, but it has also, it's known also by other names. So in the last few years, higher delta has been associated with worse outcomes in older age, both clinical health, cognitive, etc. Even, like, for example, here, survival probability over time. This and the fact that brain structure, the delta reflects brain structure differences in areas that are highly sensitive to age, it has led to the common understanding that it must reflect ongoing differences in ongoing neurobiological differences. That is a measure that is sensitive to the rate of brain aging. However, delta ends up in one way or another age residualized, so it reflects deviation from the expected norm. It's in a way akin to normative modeling, and thus it also can reflect, like, the variations of delta can also reflect differences that are pre-existing, that maybe can have, like, early life origins. So in this simplified scenario, we can interpret relationships of delta with a given outcome in two possible ways. We can interpret it as slope differences. So here is shown by these differing lines, meaning that there's a differential rate between two groups, a differential rate of brain aging or of ongoing brain changes. When you look at older populations, you will see how variations in delta are highly affected by the degree or the rate of brain change. However, it can also reflect intercept differences. So here you can see these parallel lines, meaning that essentially these two hypothetical groups would show similar rates of brain decline over time, and the differences between the two groups have earlier origins. Maybe merely they are congenital or might appear during development. You could see as well how in this embedded bar graph, how it would explain more or less as well similar rates of sensor-based outcomes, such as, for example, future dementia, future rates of dementia, for example. With cross-sectional data, it's kind of very difficult to disambiguate what or how to interpret brain age, either as slope differences or intercept differences, or both. So what we aim in this study is to use longitudinal data to disambiguate or to test both possible hypotheses. So we argue that if variations in delta reflect ongoing rate of brain aging or ongoing change, we should see a relationship between cross-sectional delta and some measure of longitudinal brain change. Here you would see how the variations, or the individual variations in the point, in like when assessed at a single time point, should be related to the slope, to the arrows. So like the change, the ongoing change, or the longitudinal change. Also, using what Stephen Smith presented before, we also argue that delta values should spread with higher age as an indirect evidence that individual delta changes over time, if it's right. And for the second hypothesis, we argue that so if delta reflects lifelong stability, we argue that we should see a relationship between delta and early life factors that are supposed to have a quite stable relationship with brain structure throughout the adulthood. Here we use birth weight and polygenic scores for brain age. And here you can see we cannot really know where the differences have emerged, if they are like already at birth or they appear during development, but they should be quite stable then when we look at them in older age. So to test this, we use the UKB biobank as the main dataset and the Lifebrain as a replication. For the training dataset, we use all individuals with a single MRI observation. And for the test dataset, we included all individuals that had 2 or more observations, so that had longitudinal MRI data. So this let us, I mean, now there are quite many more, but this led us to 1,400 and 1,500 individuals for the UKB and Lifebrain. That said, you can see the age distributions here, how Lifebrain has a longer age range, also like longer follow-ups and more observations on average, but also more heterogeneity. So that's one of the reasons why we choose UKB as the main dataset and Lifebrain as a replication. So we estimated the brain age models on the cross-sectional only datasets using a quite fairly standard and relatively simple procedure. That the main one was based on a gradient boosting algorithm, and we use a lasso algorithm as a technical replicate. We included tenfold cross-validation, randomized hyperparameter search, and we trained it on T1 regional and global T1 weight features that included cortical thickness, volume area, gray white matter contrast, and subcortical volume and intensity. So the models were applied to longitudinal datasets, so the test datasets, and show fairly comparable performance or reasonable performance matrices when compared to other studies that use traditional machine learning algorithms. If you look at and to test the first hypothesis, the rate, whether delta reflects a rate of brain aging, we use the center set of delta as the cross-sectional measure of interest. And for the longitudinal measures, we use first longitudinal change in delta. Then we use a principal component of longitudinal change by inputting, by putting all the features in a matrix. And then we also use this longitudinal change in every single feature on these 365 features. So results. So is delta associated with longitudinal brain change? First, we looked whether delta was associated, cross-sectional delta was associated with longitudinal change in delta, and we did not find any relationship. Actually, in the main analysis here, this is the technical replicate, this is the biological replicate. In the main analysis, we actually found quite a negative, borderline significant negative relationship between delta and change in delta, which it is not, that's not what we expected. And then we use equivalence test, essentially applying the same model, but shifting the null hypothesis in an attempt to reject effects of a certain or size or bigger. To sum up, if there's a relationship between cross-sectional delta and longitudinal change in delta, it is quite small, and we could reject changes that are like bigger of individual changes of 0.6 years throughout the Lifebrain dataset, for example, and 0.4 in the UKB dataset. Then, so the slides went, yeah. So then we did the same thing, but applying the replacing the change in delta by a first component of longitudinal change. Essentially, here we did not find any significant relationship here as well. And when we look at every single feature here, we did find a relationship between cross-sectional delta and longitudinal change in ventricle expansion feature, or any features that reflect ventricle expansion, because they reflect change in the ventricle. So and then finally we applied the we assess whether cross-sectional delta spreads with high rates using the formulation in the Neuroimage 2019 paper by Stephen Smith. And I'm not going through the formula, but the results show that indeed, as you show, both for the UKB and also for the Lifebrain dataset, there's a quite modest increase of delta throughout the dataset's age range that would amount about a little bit less than 0.4 years of increase in spread from throughout the dataset's age range. So to sum up this part, just delta, cross-sectional delta, is associated with ongoing brain change, but just quite modestly. I think this relates also to some of the work of that people have been doing here, telling that cross-sectional and longitudinal MRI indices tend to have like kind of very weak correlations. So next, we test whether delta was associated with early life factors supposed to have like quite stable influence in brain structure throughout the adulthood. First, we look whether normal variations in birth weight were associated with delta, and we found that indeed birth weight was associated with cross-sectional brain age delta. We found it, both, this was not published, but this is like technical and the biological replicated, so it was replicated, and the effects, the effects also survive even if we include like intracranial volume. Of course, they are bigger if we do not include this. It was also quite robust to variations of how we define normality of birth weight. And finally, we test whether the polygenic scores of delta were associated with delta or change in delta. We compute this the polygenic scores based on a GWAS analysis on the training dataset, so it was independent to the longitudinal dataset, and we found a relationship between cross-sectional delta and polygenic scores of brain age, but not with longitudinal change. Actually, it was negative, but it was not replicated later on. So altogether, this part of analysis suggests that delta reflects influences in brain structure that can have or that have early origins that maybe are already present. Some of them might be present at birth or might have some developmental origins. The variance explained is not too big, especially when we introduce intracranial volume, but as a covariate. But in a way, we consider it as a kind of a proof of concept on how better we can understand some of the variation in delta or cross-sectional delta when we look at older populations. So just to conclude and recapitulate everything. So delta reflects to a modest degree actual rate of brain change in middle and adulthood. But however, most of this variation is already present when, in the samples, when we look at the samples lower age range. And Some of it must or seems to reflect early life influences on brain structure that have either congenital or developmental origins. We must say as well that this is done in with using T1 weighted data and relatively healthy populations. So the degree in which delta will reflect or not reflect the rate of brain aging can be dependent on samples, features, modalities, algorithms, you name it. But we would end up with this caution note that it's important to have caution when interpreting brain-derived indices of aging based on cross-sectional MRI data as indices of ongoing or future brain change. This is most of these analyses are in this paper. So if you want more, you can check it out. And just thank everybody from LCBC, also the external collaborations, funding agencies, and Biobank and Lifebrain for providing the data. Thank you very much.
Thanks very much. You've brought us perfectly onto time.